I've not looked. We've not sampled systematically. But in the USA, East Coast USA, they have looked and found them, deformed wing virus in particular. There's a paper in PLOS ONE, Singh et al. With co-author Cox Foster, who I think is she is the PI at Penn State University. So there they found DWV on flower, I think on Andrena, but certainly other wild bee species. There's been one other paper out recently from the UK, a group working with Bill Hughes in Leeds. I think it might be PLOS ONE as well, I'm not sure now. Just a few months ago, in which they found DWV in wasps, social wasps. I'm not sure whether flies, but other insects, including wild bees and bumblebees, visiting flowers, suggesting that these things are quite widespread. The difference there, though, with those studies is that they find DWV, the virus. They take an insect, they crush it up, and they find DWV. Well, that doesn't mean to say that it's actually doing anything. It could well be that the insect has fed, it's in the gut, it's going to be defecated out, but it's caught and scrunched up. So there's a difference between finding it and that it's actually having an effect. In the case of the experiments here with the bumblebees, Matthias Fürst, who's a lead author on the paper, has also run experiments in which he has fed Nosema and he's fed DWV to bumblebees, and he's observed effects and recorded mortality, and they both have an effect on bumblebees. And in the bumblebees that were collected in the field, they recorded at times quite high quantities of virus, suggesting that it is actively, and also negative strand, which of the virus, because it's a positive strand virus. They found negative strand virus in the bumblebees. If you find negative strand, it means the virus is replicating, because normally it's just as a positive strand. So I think we've got pretty good evidence that it's quite widespread, and it's doing something in bumblebees as well. But for other bee species, we don't know, but we've not looked.
So do you think that these viruses are usually spread, or they were in insects somewhere in Europe before, or it's really new?
The virus DWV is not new.
Yeah.
It's been found, I think it was first described in the 1960s. And apparently 1 in 10,000 honeybee colonies used to have DWV, or was found in it. So the fact that it's found in a variety of other insects also suggests it's probably not honeybee specific. Maybe its natural host is not even honeybee. But the varroa mite on the honeybee seems to have led to its amplification in individuals that are infected with, that are bitten by the varroa mite. Either because the varroa mite is suppressing the immune system, or whatever system, the innate immune system, or whatever system the honeybee has for suppressing viruses. Actually, I don't think it is that. I think it's simply that the varroa mite is introducing a lot of virus, and the virus then can amplify, and the bee cannot, its antiviral system cannot cope with so much virus that's getting in, I think, actually. But that's just my own feeling. I don't have data on that, on the varroa issue. Yeah. So probably widespread, and it's widespread in North America as well. So it's probably a general insect virus. It's my suggestion.
Maybe the cause of the colonies. Do you think that it's really connected with these viruses, or could be there something else, like mobile phones or whatever?
Well, I'm working on the viruses, so I probably will say, no, it's the viruses. I know that many people have suggested, there's been a lot of discussion about pesticides having a role, GM crops having a role, lack of floral resources, having a role for honeybees now. We have run, and particularly Vincent and Maureen, two postdocs with me, have run umpteen experiments in which they've given viruses, or microsporidia, and also pesticides, sublethal doses of pesticide. And they sometimes find an interaction, and sometimes they don't, with the pesticide and the pathogen. So actually the impression I get after looking at a lot of experiments is that the viruses are the major factor. And not those others are responsible maybe for a small % of the mortality. My feeling is, after looking at all our experiments, is the main reason, 80–90% of that decline is the virus induced. But I'm going to be biased because that's what I'm working on. Yeah. So, but I would suggest so. And you might say, well, why is it that it's a problem now and wasn't a problem 20 years ago? Because Varroa has been around for quite some time. And I think part of that has to do with the way in which Varroa is controlled. 20 years ago, this exotic Varroa mite was not so frequent, and when beekeepers had it, they controlled it using a variety of chemicals, particularly pyrethroids. And they could kill 99% of the mites. Now, the mites have evolved resistance to those pesticides that are applied by beekeepers. So when a beekeeper goes and treats a colony, he's not killing 99% of the mites nowadays. Maybe it's killing 80% of the mites. So the parasite burden remains quite high. And I might suggest that what's happening is parasite burden remains high, and the virus therefore remains high as well. And it just builds up further. So the beekeepers aren't able to, at least in the honeybees, they aren't able to knock the mite far down, which would also bring virus levels down. So I think that's why in the last 5 years or so we see more of a problem. Beekeepers see more of a problem. That's the case over here in Europe, and it's the case in North America as well. The common chemicals which used to be used to kill Varroa mites by beekeepers that are not so toxic for bees but very toxic for mites, they don't work so well anymore. And I think it's just that general phenomenon. That's my take on it.
Maybe I missed something. Have you any explanation for this decreased polymorphism in parasitic?
Yes. I don't work on Hymenoptera parasitica, so when we looked at the data, I was expecting to see a nice signal of haplodiploidy and eusociality, and then I saw this, parasitica data was also showed very low diversity. I will put that down to inbreeding. And many of the species for which the microsatellites have been developed are inbreeding species, and they're used also commercially. So maybe they go through bottlenecks, genetic bottlenecks in their rearing, and that's why they show relatively low genetic diversity. I don't really have a good explanation other than that. We do know that the Hymenoptera parasitica, many of them undertake inbreeding. Clearly they have a mechanism of sex determination which is not complementary sex determination. We know that. They've got genomic imprinting, works in, I think, Nasonia. I think I've got it right, Nasonia. So they've got other mechanisms of sex determination. But many species, not all, but many species are highly inbred. And I think that may be an explanation for why they have low genetic diversity in breeding, which would make sense.
Are there any related data on other groups of parasites?
No, in the data that we looked at Coleoptera, Lepidoptera, Diptera, and Hymenoptera, those are the 4 major insect orders with most insects in them. We didn't look at other parasitoids or other. You asked about parasitoids.
I mean another groups of parasites, not just parasitic insects. Whether this phenomenon was observed also in some parasites.
What I think would be useful to look at is other haplodiploids. There are thrips, there are some beetles, there are whitefly that are haplodiploid.
But they are not parasitic species.
No, but in terms of the hypothesis about haplodiploidy, one could look at those. And the problem is I don't think there are many species. I don't know whether there are any microsatellites developed for them. Maybe there are. We have looked at, so one hypothesis that we had is haplodiploidy. The other hypothesis was eusociality. And we have looked at the termites, which are not an order anymore, but they're anyway nested within cockroaches. And the termites show reduced genetic diversity, they're diplodiploid, compared with other diplodiploids, which is what one might expect because of eusociality. So there is an out group, if you like, that we've compared, but we've not looked at other parasites or parasitoids.
Is genetic part? If I remember, there are not all species with complementary sex determination have only a single locus. It should be polymorphic. In some primate species, there are several also. So have you checked whether your species, like Scorpion species, have just a single locus, sex determinant locus?
Yeah. Yeah. Now, you're right, we don't know whether it's single locus or multi-locus complementary. It's clearly complementary sex determination because we sometimes get diploid males, but most of the time haploid males. So it's some form of complementary sex determination. The only way to tell really for certain is to do mating experiments where you get a male and a female, brother, sister, and mate them, and there you'd expect 50% of the offspring of a matched mating to cause if it's single locus. What I will say is that those sorts of experiments have been undertaken on, I think, 7 or 8 other bee species, and they all have single locus complementary sex determination. So the honeybee and other species that have been looked at have a single locus. Of course, there's 20,000 bee species, and it's 6 or 7 may not be representative. Where multilocus has been found, it's I think in the Parasitica. There's a good example of I think it's Gonionus, but I don't recall now, is of a Parasitica which seems to have multilocus sex determination. And I've worked a little bit on some communal nesting bee species, Andrena, Scotica, and there, I get a lot of, genetic data suggests a lot of inbreeding, but very little diploid male production. So maybe that species also has a multilocus system of complementary sex determination. We don't know here. So it could be that orchid bees also have multilocus. Yeah, it's a caveat of that we are under the assumption of single locus. Yeah.
I also wanted to ask on this diploid male production, because in my analysis on Host-pathogen, we discovered very little genetic diversity in Japanese populations. There are 2 or 3 illnesses per fellowship, generally. But we haven't detected any diploid males, so which was very surprising for me because I know, for example, they describe in Host-pathogen or introduced species Jistě, and we discovered a very large number of diploid production in the species which was introduced in general numbers. So
Yes. I mean, the model explaining the diploid male extinction vortex assumes, and there's an important assumption there, that the reproductive rate of females is quite low. So if the rate of reproduction, the numbers of offspring they have, is quite high, which is probably the case in that species, actually they can breed their way, if you like, out of the extinction vortex. They might produce lots of diploid males, but they're producing lots of diploid females as well. So the population doesn't go into the vortex. And you're right, there's a strange example. It's Laciglossum leucosonium, is that right? I think it's the species that's been introduced to the USA. It's got diploid males, but the population is expanding. And it's the same in Tasmania with Bombus terrestris. It's been introduced, probably from a founder event, very low genetic diversity, low sex allele diversity, lots of diploid males are produced, but the population's expanding. So presumably they can overcome if they have a high enough rate of reproduction. So that's one of the features of the model, the theoretical model. It's one of the fine details, mathematical fine details. Low rate of reproduction leads to that extinction vortex. So what did you say? Did you say you found very low variability?
I found none that diploid males in the population, which was very drastically genetically reduced.
Oh, of which species? Oh, Anthrophoplumipes. Oh, right. Uh-huh.
Of course, we analyzed about 50 females only, but still I expect to find that reduced.
Oh, right.
Quite interesting.
Yeah, it is, isn't it? That's strange, yes. I must say, we're working on another data set of another orchid bee on another island, one that seems to have much more a viscous population structure that's been described. And actually we get quite a lot of diploid males. So sometimes it does happen. But I think for most orchid bees, their population size, NE, is quite large, actually. And they may even have overlapping generations, which helps to enhance NE, so they're long-lived. And I think, actually, the orchid bees, at least, don't seem to suffer so much. They can fly very far. Females are known to fly several kilometers over open water, I think up to 5 kilometers. Okay, our island was 11 kilometers away. 11 kilometers is quite a lot of water. But they have, some individuals have been observed flying 5 kilometers. So they are obviously quite tough, strong flyers, and therefore some degree of habitat fragmentation may not actually impact upon them for orchid bees. I think for other species, I'm sure it probably does, because it's very species specific.
Do you know what could be those reservoirs of those viruses in other animals, let's say? Okay, let me add.
Yeah.
Here in Czech Republic, there are used some acaricides, chemicals against those varroa mites, which have been, or maybe still are, used in Britain against mites of sheep or cattle. So my first, let's say, idea would be that maybe those mites are at least very similar to some mites which suck on cattle or sheep
You're right that the acaricides that are used or have been used by beekeepers are also acaricides used by sheep farmers, because the acaricides are attacking often they're attacking the nervous system of the mites. So I can imagine there would be commonalities between mites on sheep and mites on bees. Varroa, though, is pretty specific. It needs to go onto a honeybee pre-pupa to be able to initiate oogenesis. And it won't even do it on an adult. And it won't go onto a bumblebee. I don't think they've ever been able to successfully rear varroa mites on bumblebees. So it really it's quite restricted in its host choice. It's very good on that host, but it's quite restricted. So, if it doesn't go even onto bumblebees, which are somewhat closely related to honeybees, and probably not on other bee species, the chances that it would go onto a mammalian host I think it's 0. Effect. And vice versa. I think mammalian mites, I don't think we've ever found them on bees, or bees carry a whole range of different mites, one should say. So there are actually a lot of potential vectors out there. The honeybee has got this exotic varroa mite, but bumblebees have, I don't know how many different mites, 10, 100 different species on them. I can't. It's somewhere between 10 and 100, I think, have been described. Let me put it that between 2 and 3 orders of magnitude. 1 and 2 orders of magnitude. And other bee species have got their own mites associated with them. So that many of those, and I think that's the case probably for many insects, they have got potential vectors of pathogens. But I don't think there's any sharing between vertebrate and invertebrate. Not that I'm aware of.
sperm, and are they mating with a female? If not, so then it would be quite easy to eliminate them because there could be in future multiple matings.
Okay. In the case of the honeybee, the diploid males that are produced, they are produced, and occasionally, if the queen has mated with a male that carries the same sex allele, it happens, and it happens probably quite often as well. But just at if she's mating with 20 different males, maybe one of those males is a match mating. So a 40th of the diploid larvae are diploid male. They are eaten in the first 24 hours after hatching. So she lays the egg. It's a diploid, but it's recognized by workers and eaten straight away. So the colony never actually produces any diploid males. One can rear them in a laboratory. If one takes the eggs and rears them with, or when they hatch, rear them with royal jelly, they'll develop into diploid males. And people have done that, and they produce sperm. But it's diploid sperm. And if that's inseminated into a queen, I'm not sure whether they've managed to get triploid females, but I think they're sterile then. So that it is a genetic dead end. It's been suggested that that the diploid male larva, in 24-hour-old larva, may produce a pheromone to advertise that it's a diploid male, because it's been kin selected. Suicide has been kin selected in the honeybee so that he doesn't waste his colony's resources, if you like. It's a kin selected trait, which seems reasonable. Kin selection may operate in that way, certainly. For other species, and I think there might be the case for one bumblebee, and certainly for one bumblebee, triploid females have been found. And I'm also working on a data set of it's actually a Patulapis species. It's a sweat bee species from South Africa, Doloritica, which is a rare species. And we have found, in analysing, I think about 300 individuals, of which about 100 are male, 1 or 2 diploid males, but we've also found a triploid female. And another group in Scotland, that's the group of Dave Goulson, have found I'm not sure where they found diploid males, but they have found triploid female workers, suggesting that there must be some diploid males around. So occasionally, in small populations, these things are found. Yeah. I mean, one of the problems is that if diploid males are inviable or have low survival, the chances of us finding them are low. Even though they're being produced, we may not catch them. But it's interesting that in this Patulapis that we have from South Africa, and in the bumblebee from one of the rare bumblebee species in the Scottish Isles, Western Isles, I think 2 or 3 triploid females have been found, suggesting, well, like some populations may be suffering then from that. Yeah. But not orchid bees. The impression is, anyway, from our analyses, they probably have very large population sizes. There are other species. There's one very famous wasp species, Odoneus, in the States, that has complementary sex determination, but is a regular inbreeder. So that something like 80 or 90% of the matings are brother-sister matings. And there, the diploid males are viable. And they produce, I think they produce haploid sperm. Have I got that right? Cowan and Stahlhut, 2008, I think, PNAS. It's quite a remarkable situation. So for species that seem to regularly inbreed, they may have evolved an ability to cope with this complementary sex determination. So there's an example of a single locus complementary sex determination, but they've overcome the issue of diploid males. I can't now recall whether they're making diploid males are viable, and they mate with females. And I think they must undertake a meiosis, I think. I don't have the details on the top. But it obviously is not harming the population, so occasionally it happens. But I think for the vast majority of these, the things that we're interested in, the bees, the vast majority of them are regular outbreeders, probably with single locus complementary sex determination. And therefore, probably, if there's a match mating, the diploid male is a rare diploid male, and it's inviable or sterile. Yes, ants were included there as well. So when we looked at that, it's not really a formal comparative approach, but when we've looked at that cross-species approach, then ants are included as well, and there's a lot of work that's been done on ants. So, but if we look within the eusocial Hymenoptera, the ants are no different from the bees or the wasps. You know, I thought, oh, maybe ants are low and honeybees are a bit higher and bumblebees are a bit higher, but they seem to be all mixed in together there. Yeah.
Are there any evidence about the possible role of global warming in this change? I mean, for example, the difference between mountain species and lowland species?
We were talking about this yesterday. In terms of species distributions, yes, that certainly is the case. And there's been a lot of discussion of how that might impact changes in species distributions and abundance might impact host-parasite relations. In principle, it could do so. I don't think I've seen any data, at least not for insects or not for bees, where that's been suggested to be the case. So, but maybe there is, so it's an interesting question, that. Yep. So we can think about other cases of emergent pathogens, like critid fungus attacking amphibians. Now, in that case, amphibian populations worldwide seem to be suffering from that critid fungus. I'm not sure whether global change, climate change has been suggested. In that case, it seems to be a new recombinant or a new more virulent fungal genotype that seems to be causing the problem. In the case of the white nose syndrome of bats that I was talking about 2 days ago with one of your colleagues, I think it's unclear there what the situation is in the USA, where the phenomenon is found, whereas it seems over here in Europe we don't seem to suffer, at least most bat species don't seem to suffer. And the genetic diversity of the fungus is much broader as well, is that right? So I think in the US it's a very narrow, it's a very single or single genotype, is it? Or a single variant, genetic variant that's causing the problem. But it's instructive to talk to your colleague, because it's not clear the issue, and maybe there's some relation with climate change for those diseases, for deformed wing virus and VDV, maybe. It's difficult to tell. You know, in the case of honeybees, they maintain a homeostasis in the colony. So inside the colony, it's 35 centigrade. Summer and, well, not winter, but at least when they have brood in the colony, it's kept at 35 centigrade, 70% relative humidity, 1 to 2% CO2. So it's fairly stable in that case, whether they're in the desert or up in the Arctic in summertime.
But in different temperature, it can be much more expensive for them to keep temperature. Yeah, no, that is true. Yes, yeah. So I think that I would say that's an interesting open question. I was impressed by this difference between eastern and western part, because I think it's the mountain part and the lowland part.
Yeah, there is mountain on the east in Ireland. Do you mean? Yeah, there's mountain on the east, but on the east there's also arable agriculture, and on the west of Ireland there's no arable. Maybe some potato, but it's otherwise sheep grazing, cattle grazing, and tourism.
He has several subspecies.
Yeah.
Are there any data how they survive, or how they handle similar pathogens, or maybe the same?
I've only seen one experiment described looking at honeybees in Spain, where there are supposed to be two different mitochondrial haplotypes, a North African or an African haplotype and a European Apis mellifera mellifera haplotype of mitochondrial DNA. And there I've seen one experiment, a set of observations, where they've looked at the instance of nosema in the two different types, and they seem to suggest that one of the types in 1 year had a higher instance of nosema than the other haplotype. But in the second year, they didn't see that effect. So that's the only bit of evidence that I've seen to suggest there might be variance there. My feeling is that maybe there might be some differences amongst races, subspecies as they're called, but I would guess within a subspecies there'll be a big variance in response to pathogens. So, you know, if one's thinking about breeding honeybees that are resistant, probably the best thing to do is to look wide enough within any one race. So if you're here, look in Apis mellifera carnica, if that's what's here, or and in Italy, look in Apis mellifera ligustica, and select within that genetic stock, probably. Rather than move honeybees from one part of the world to the other, because that's why we're in the problem that we are now, because of humans moving things around the world, including moving honeybees and inadvertently moving varroa mites. So we're the root cause. I don't think the bees ever cure themselves of nosema, because the nosema is inside the ventricular cells, inside the epithelial cells of the ventriculus. And once they get in, they destroy that cell. And then they shed either into adjacent cells or back into the lumen of the ventriculus. So I don't think a honeybee can ever cure itself of nosema. It can, though, nonetheless fly out and defecate outside. I will say, let's see, so there's several things I'll say here. One is that I think that Nosema serrani is a pathogen. It does reduce mortality, it does reduce longevity of bees. So I have a close colleague who argues against that. He said, it's not doing anything at all. It's not even a pathogen. Maybe you can consider it a commensal. I think it does have, it is pathogenic. It's not very pathogenic if the bee can fly. Our experiments, though, indeed, all the experiments that I've described to you, have all been undertaken in the laboratory in cages, in a highly controlled environment. And that's a caveat of those experiments. We've not got the bees in the full colony. We've got them in little cages. Each one has been individually fed or individually injected. So it's a highly controlled experiment, so we can be really sure the effects that we're seeing are caused by our treatment. So that's the advantage of the approach we take. The disadvantage is that, well, we don't know that's exactly how they would be in a real colony, indeed. So which is, I think, is your point. And I've got to say, indeed, it might be that these pathogens have different effects in colonies. The PhD student Massini, with me in Halle, has been looking at that, though. So she's been doing it exactly the same, but rather than putting bees in cages, putting them into an observation hive or into full hives and then recording their behavior. And the impression we have, and she's just analyzing those data now, vast amounts of ethological data, the impression we have is that the virus changes behavior quite considerably, and I think also physiology. The nosema doesn't seem to, it shortens life maybe a little bit or maybe not at all, but it doesn't seem to have such a strong effect. The virus, though, is really changing behavior. And I think my take on it, so I don't know, but my take on it is that it completely changes the physiology and the behavior of the individual. So it's more or less redundant. It's not even working. That's my take. So I don't know that that's happening, but at least from the behavioural data in full colonies, it really has an even more profound effect than we see in the cage experiments, actually. Nosema may be slightly less. Yeah. Yep.
I wonder whether the individual bee workers did not die just because they were, let's say, separated, because there were too few in the group. I think that if you just take one bee worker out of the beehive, it dies in an hour or two.
Yeah.
It just has no contact with others.
Yeah.
So I know that you showed on the slide that you get 25 or 30.
Yeah.
Did you somehow, how do you come to this?
Okay.
And would it be better if what was the mortality?
Compromise. It's a compromise because we want to ensure statistical independence of the data. So when we have an experiment, we take 5 cages per treatment. We try to take 5 cages per treatment, and maybe I think it was 21 bees, I said, per cage. And we found that 21 bees they do okay together. 21. Each bee is treated individually. However, each bee is in a cage. So when we run our analyses, we have to take cage as a random effect, because the cage itself there could be differences between cages. So to be really strict and thorough about the statistical analysis, we can take individuals, as they're not truly independent because individuals are nested in the same cage. But cage is a random factor. Now, we actually take bees from several colonies and we mix them amongst the cages. So we've not got a colony effect. We're not looking at genetic differences, because that would just compound the issue further. So we try and obviate genetics in our experiments, so it's not a statistical factor, because the bees are randomized, actually they're equalized across cages within a treatment and then across treatments. Why 21? Because we find with about 20 bees they do quite well. They survive till 40 days in a control. And so if we have fewer than that, actually what I would prefer is that each cage has a single bee, and then have not 20 times 5, but 100 individual bees, because then I could treat each of those statistically independently, although they would actually have come from just a few colonies probably, so there'd still be a blocking factor there of colony. But it's a statistical problem, and we want to ensure that we have enough replicate cages to be able to make have a reasonable statistical power. So if we put them all into one cage, which is another thing we could have done, we could have put all 100 into one cage. We've got n equals 1, we've got no replication there whatsoever. So, if we get a higher mortality in that cage, is it because of the cage or because of the treatment? You can't tell. That's why we have multiple cages per treatment. And hopefully you've noticed that when we have 5 cages of the virus treatment, 5 of Mycobacteria, 5 of mixed, we also have 5 control. So we have as many control cages as we have these others, and that's absolutely necessary as well for to get a proper variance estimate of the control as well as the cages. It's purely compromise. It would be nicer to have more bees per cage and more cages, but it's a lot of work individually injecting and individually feeding every bee. And it's simply that compromise between statistical power and time and effort and money.
It's a nice difference. So, is there any important question? Final important question? We usually continue with discussion till everybody is exhausted, which is now like this. So thank you very much.
Pleasure. Thank you. Thank you for your attention.